I'm Dr. Sean Coakley from the Queensland Brain Institute at the University of Queensland. I'm a neuroscientist, which means that I study the brain. I'm interested in how the brain is built, how it's wired together, and importantly, how it stays together for our lifetime. Our brains contain billions of cells, called neurons, which communicate with one another via electrical signals sent through long, thin, cable-like structures called axons. Just like the cables in our homes, axons need to be protected in order to work properly, but they're very thin, only 1/50th the width of a human hair, which makes them very vulnerable to damage, which can have devastating consequences for the ways that our brain works. My research is focused on understanding two of the deepest and most general of biological questions. How do we keep an axon together for our lifetime? And what can we do to delay or prevent damage to it when injury or diseases strike? I've discovered that the tissue around an axon is really important for keeping it together. If we can understand the molecules that are important for this protection, we might one day be able to slow down, prevent, or even delay damage to the nervous system caused by injuries or disease. Well, I think that every day people would probably think that I do some pretty weird stuff, and that's mostly because of the organism that I work with. I work with a very unassuming animal, a tiny microscopic nematode roundworm called Cenorhabditis elegans, or C. elegans for short. This is a tiny microscopic worm that has a very simple nervous system, which makes it very easy to study the brain. It has only 302 neurons compared to the 100 billion that we have in our brain. And also, it's see-through, which means I can just peer inside and watch what's going on. We can also freeze these animals in a kind of transient hibernation state, and then we can just defrost them when we need them, which is super convenient. One of the best things about them is that they're very amenable to genetic manipulation, which means researchers like me can tinker with the DNA and make tailored changes to the neurons themselves. The idea is that we could deduce bit by bit what each molecule is doing by getting rid of it and seeing what changes. This is a painstaking and laborious approach, but it can often have really breathtaking advances in our knowledge.